Hello, everybody. I am Audrey Dubourg, Product Manager at CN Bioinnovations, and today I'm going to talk to you how to address both sides of the coin and how microphysiological systems can help better predict safety and efficacy of novel therapies. So currently, scientists have access to a wide range of tools in their preclinical toolbox to evaluate new therapeutics and molecules. They usually start with in silico assays to really find the right targets and the right compounds. Then we move on to 2D cell culture and 3D cell culture in vitro to screen and test, and then when we have more insights, you can go into the in vivo animal studies before moving on to clinical trials. However, all of those toolbox currently raised one question is how do they translate to the human, and can we make sure that what we have as a preclinical data actually works in the clinic? Unfortunately, currently, most of those approaches lead to therapies and molecules failing clinical trials 90% of the time. We therefore need new approaches to develop in vitro assays. When developing those assays, we do require a lot of different factors to consider to really input into those assays. And some of those are first about how reliable and robust the new assay is, what is the cost, is it viable in terms of budget to use the assay and implement it, can clinically relevant or translational endpoints be used as well in the assay? What is the throughput? One obviously big important question is it representative of the human biology? And when working with drugs or compounds, we also need to ensure that it mimics the in vivo pharmacokinetics. We need to make sure that we can do acute and chronic dosing. Can you assess the population viability in that to have more insights? Can any immunological effects be added as well to the assay? And with the trends of new modalities being more and more being developed and used, such as ASO, so gene therapy, can we study the effects of such modalities in those new assays? So one new technology being recently developed in the last 2 decades are microphysiological systems, also called MPS, and they usually come in your preclinical toolbox after you've used 2D culture and 3D organoids and before moving into the in vivo animal studies. What are MPS? They usually are defined by being 3D cell cultures, typically using human cells, and with the fluid flow used to mimic the circulatory system, either gravity-driven or using true fluidics in the system. How do those compare to the standard methods currently used in your preclinical toolbox? What we know is that humans usually have human cells. They are composed of 3D complex organ tissues. They have a blood flow connecting these organs, and also inside these organs, you do have an immune system, and therefore you can study human drug exposure. So clinical trials ticks all the box to try to work the drug discovery. Animal models like mouse do not offer human cells because they're different animals, but you do have the complexity of a whole system and organ, and you do have a blood flow between those organs. However, the immune system is very different, and therefore to study the human drug exposure is very limited. It is also very slow to get results because you have one condition per animal. Conventional assays are a bit faster in terms of results, and they do use human cells. However, most of them often offer immortalized cells, so they are with the phenotypes very changed from the original organ they target. But you do not have a 3D complex organ tissues, neither you can have a blood flow between those organs. The immune system can be sometimes added, but it's still not fully representative of the human, and the human drug exposure is therefore not fully assessed in those assays. MPS are quite different because they do offer proper human cells. When I say proper, is primary or iPSCs. You do have 3D complex organ and tissues. You can connect different organs together to have a blood flow between those organs. The immune system can be also added, and therefore the human drug exposure, when you assess it in those systems, is related to the human and the clinical data. And also the time to results is also quite fast because you can have data and answer within 2 weeks up to 1 month when running experiments. So before I get into really the applications from this talk today, I'm just going to briefly introduce CN Bio and what we do and why I'm talking to you about that today. So CN Bio commercialized MPS devices, and our system, the PhysioMimix, was actually born out of a technological race that was sponsored by the US Defense called DARPA between MIT and Harvard, and we were part of the MIT consortium to develop 7 up to 10 organ on a chip. And that race was won by a consortium back in 2016. And in the back of that, we developed our system, the PhysioMimix, which was launched in 2018. And ever since its launch, had been used with the FDA Center for Drug Evaluation and Research to really assess and evaluate the use of organ-on-chip system in regulatory submissions. And in 2021, we launched our first multi-organ PhysioMimix platform with application in ADME and toxicology. So here we see the system. I'm just going to briefly introduce it to you as we're using that system for all the different applications covered today. And we can see on the left-hand side a big white box with a touchscreen that is a controller, usually sits outside of the incubator next to it and controls all of the biology that happens inside of the incubator. And that biology is performed using the 3 plates you see on the right-hand side of the controller here, which are open well architecture, and the flow is driven by the microfluidic drivers on which they are connected to enable total perfusion of the biology inside. The current platform offers 3 different types of plates, different to develop different organ models. We do have a Bio plate, the Transwell T12, that is used to develop bio models such as gut, lung, or skin. The liver chip plates, which is used to develop 3D liver models of either diseased or healthy liver. And we have a Transwell liver that offers a 2 organ plate to study the interaction between the liver and a bio tissue such as the gut or the lung. So the experimental design works similarly in such platform as any kind of cell culture. You first decide on the cell format you want, let it be cell lines or primary cells, stem cells, or even commercial models. And you're going to seed your cells or put your tissue in there, wait for the tissue to be ready, and then start either to induce the disease states or do repeated drug dosing over time. And we have gone over 30 days using our liver model, and the data are generated from the culture media, the tissue structures, and if you need to perform some non-invasive measurements, probes can be added into the system. All of the data at the end analyzed using all the typical endpoint analysis from omics to microscopy, histology, biomarkers, and metabolites as well. So now we have done a bit of an introduction on the system. I'm going to talk to you about different models of liver MPS used to better predict safety issues in the preclinical data. So one of those liver models was developed in-house, and we used our liver plate, the LC12 you see on the left-hand side there. That plate inside of its well has what we call a 3D liver scaffold. It is on the bottom right-hand side of that slide next to the images, and that scaffold has microchannels in which the cells are seeded in the well, and then the tissue is going to be created over time from single cells to microtissues via morphogenesis, and using a flow that actually circulates throughout the 3D tissue, enabling all of the cells inside of the 3D structures to access nutrients and oxygen. The tissue is forming tight junctions, so the hepatocytes are actually polarized, and we also have bile canaliculi that are formed, recreating the liver microarchitecture in the plate. We can see here in terms of the bile canaliculus, we do have a formation of microvilli inside, and the lumen is also there and around the tight junctions, really showing the microarchitecture of the liver inside the plate. The tissue is highly functional for a long period of time. We can see on the top graph on the right-hand side that albumin is secreted over time and quite steadily even after 28 days, whereas LDH that shows cell death is quite low, showing that tissues are healthy and functional. They also synthesize a lot of different proteins like fibrinogen, A1AT. We can measure the expression of a full range of different CYP enzymes, and that's the graph on at the bottom. You can see on the right, where we see a lot of different CYP enzymes being secreted, and we can see the really metabolic activity and the donor-donor variability when using different donors. The model is transcriptomically stable, and we do have transporters that we can assess in the model. The model can be made a bit more complex, adding non-parenchymal cells, so we can add Kupffer cells or stellate cells, or even all of them together to have a really complex representative model of the liver. And by doing that here, we've tagged the Kupffer cells and stellate cells with EGFP and mCherry just to ensure that we could see them in the tissue, and we can see that they're all present in there. And when running transcriptomics, we can see that the three cell types all have the markers there, so they're all present in the cell culture when seeding them together. We compare that model to the gold standards currently used in pharma companies, which are spheroids and 2D sandwich cultures. And what we see on the left graph for the metabolic activity, the liver MPS, which is in blue, has a much higher and much stable metabolic activity for longer period of time when compared to the sandwich culture, where in yellow we see that after 12 days we have no more metabolic activities, and spheroids have a steadier, longer metabolic activity, but still much, much lower than what we see in liver MPS. And we observe a similar trend about albumin production, where we have a much higher production of albumin in liver MPS for longer period of time compared to the sandwich culture and the spheroids. So we use that model internally and our collaborators as well to assess drug-induced liver injury, DILI. DILI is a very big issue when developing drugs. Most of the time it is detected too late, usually in the clinic or even when a drug reaches the market, which is why we need better in vitro assays to really assess those risks before we bring those drugs to the market. Here we have the example of troglitazone. Troglitazone was actually released into the market in 1997, but was withdrawn 3 years later due to some undetected DILI in patients. So we wanted to see if we could recreate the DILI event for that specific drug in our system. And for that, we used a co-culture of hepatocytes with Kupffer cells. We did chronic dosing using multiple endpoints from CYP activity, ATP, LDH, albumin. We also used clinical biomarkers here, ALT. We generated IC50 and EC50 curves to generate an hepatotoxins product distance signature across those multiple endpoints. And what we can see from those graphs is that we do have a dose-dependent DILI event happening with troglitazone, where we lose the metabolic activity, the cells are dying because ATP is decreasing, ALT and LDH are increasing, and we also have a loss of function of the liver tissues because albumin and urea are also compromised with it. So we really have a good understanding of what the DILI event is for that drug. We can also, because of that, create a bit of a table and decide and determine the actual DILI concern for drugs using those different assays. And we can see here on those graphs that the assay is sensitive enough to really detect low to mild levels of hepatotoxicity on top of the high DILI concerns for drugs that are well known for it. So we can really understand what the risk is in terms of safety for those new drugs ahead of time. Those liver MPS also allow to dig deeper and try to understand what are the mechanistic details behind that DILI event. And that is done because we can measure functional CYPs, transporters, and also the bile acid machinery. And all of those enable the detection of reactive metabolite toxicity, mitochondrial perturbation, cholestatic injury, and also transporter-mediated toxicity. So here we can see the graph on the left-hand side. We can see diclofenac toxicity over time at different concentration, and we see mitochondrial perturbation and cholestatic injury from that graph. We can after that use broad omics approach to assess the toxicity profile, and here by using proteomic screenings of secreted proteins, we've identified that the majority of proteins secreted were associated with apoptosis, 66% of them. So really tease out the difference between different compound sets and the DILI event and the cause of the DILI for such drugs. Another type of safety assays that is quite key to have better model is genotoxicity testing. And that is because currently the assays offer for that are run in vitro and in vivo all in rats. They do provide high false positive rates, which is really tricky to interpret. Therefore, there is a poor correlation with clinical genotoxicity data. So here the work I'm going to present was actually developed by our collaborators at Charles River, where they developed a multi-cell type genotoxicity model incorporating liver and immune cells using our system. And for that, they actually used the Transwell plate, also known as the Bayer plate, in which they used a sandwich culture of liver endothelial cells, LSECs, and hepatocytes at the bottom of the well to be directly exposed to the flow. And in the transwells, they actually added human lymphoblastoid cells, so immune cells, TK6. And for those two different types of cells, they have used two genotoxicity assays that are internationally recognized and part of the OECD assays that are mandatory when assessing DNA damage. They use micronucleus evaluation on the immune cells, and they used COMET assays to study the DNA strand breakage on the liver cells. So they used for the liver model either primary hepatocytes or hepa-RG, but here I'm going to present the data on primary human hepatocytes. So what they've done is to assess genotoxicity in those liver models. They used indirect mutagens that are well studied, benzopyran, BAP, and cyclophosphamide, CP, and they also used direct mutagen metanesulfonate, MMS, and ethyl metanesulfonate, EMS. And what we see here is that we do observe for the liver tissue on the top graph really an increase in genotoxicity with those drugs that we couldn't observe in the rats. And we also see a similar effect for the macronucleus in evaluation on the TK6. And all of the data they showed here actually match the clinical data for the compounds they used. So really showing a good correlation between these liver MPS models and the clinic. The other side of the coins I mentioned earlier is to better predict efficacy. So here I'm going to present two types of MPS models. First, our liver model for disease modeling, and then a lung model, which is quite trending with the pandemic that has been hitting us for the last 2 years. So the liver model we used was performed in-house using our standard liver plate, in which we used human primary hepatocytes, Kupffer cells, and stellate cells, and we recreated non-alcoholic fatty liver disease in the plate by using different fat and sugars in the media. And we created the different stages of the disease to really be able to study the therapeutics of interest in there. NAFLD being a huge issue in the Western world with the Western diets nowadays. It is something that still has no treatment because most of drugs failed in efficacy clinical trials as well. So how to model NASH, we use a triculture with the 3 cell types I've just mentioned. We can actually measure the inflammatory profile, and we see that the NASH model has an inflammatory profile that is maintained throughout 2 weeks. We can see a wide range of cytokines being also secreted over time, and we see a difference when it's only steatosis, which is hepatocytes only, versus the NASH phenotypes with the 3 different cell types. We can also measure fibrosis in that model when assessing NASH, and we can measure the different biomarkers as well expressed, so fibronectin, TIMP1, or alpha smooth muscle actin. So we really see a really nice spectrum of the whole disease in the plates. When comparing this model to the current model available, on the left-hand side, we can see that our model really compare much better the human gene atlas to um, compared to the other models currently accessible, whether let it be in vitro assays on human immortalized cell lines or actually rats model. There is also a close association between human NASH and our model. We can see in the Venn diagram on the right-hand side on the top that we do have much more genes associating with the disease from human to the MPS compared to the murine model. And when comparing the expression of key genes associated with human NASH, we see that our MPS model matches up to 50% of NASH patients compared to murine model going barely to 10%. So there is really a need for those models to study disease modeling. So once we've established that model was good enough, we wanted to see whether or not we could recreate the efficacy of known drugs that were in clinical trials, so elafibranor and obeticholic acid, also known as OCA, and we recreated different dosage for the two drugs to see if we could decrease the fibrotic phenotype in our model. And what we can see here on the microscopic images is we have a dose-dependent reduction of the actual fibrosis, and we can measure that after that and quantify it in the liver microtissue, showing there is an effect that actually matches the findings in phase 2 clinical trials for those drugs. Another model that has been quite popular and greatly needed in the last 2 years was to develop lung MPS models. So here I'm only going to present the alveolar model, but we do have the broncholar model as well. So what we've done here is using our bioplates, using transwells, we have seeded in an air-liquid interface multiple cell types of the lower airway, including 81 and 82 cells, and we cocculed them with endothelium cells, and the endothelial layer is actually seated on the basolateral side of the transwell to be directly exposed to the flow in the plate. What we see from there is we do have a nice 3D-like tissue structure for endothelial layer due to the exposition to the flow, and even our epithelial layer has a really nice depth and ciliage here, and everything looks like the lung, really. The biointegrity is also much better when the two cell types are co-cultured together compared to their monoculture counterparts, as you can see on the bottom graph. And more importantly, we measured the functional markers for 81 and 82. As those two cell types tend to differentiate in vitro, we want to see if we could maintain the phenotype in that setup. And what we see is we do have still those two cell types in the lung model, really recreating and maintaining the lung phenotype for the alveolar model. So we used that model to study viral infection. We could not go without studying SARS-CoV-2 infection, as it's been on everybody's mind for the last 2 years. So the way we've done it, we used a pseudotyped lentivirus expressing SARS-CoV-2 spike proteins, and we infected on the apical side. We ran a 48 hours infection period. We sampled media of a different time points, as you can see on the timeline, and at the end, we ran microscopy to see how infectious that was. What we see is that after 48 hours on the microscopic images, you can see that the bottom images do show infections with SARS-CoV-2 pseudotype lentivirus, where we see all the red in the merge images are actually infected cells. We've also observed an increase in disease-specific cytokines, where we see when you have the lentivirus there, we have IL-6, IL-1 beta, and TNF-alpha that are actually increased over time compared to the mock, where there is no viruses there. And those cytokine profiles actually do match COVID patients, and therefore the model mimics COVID-19 patients profiling for cytokines. Once we've established that model, we wanted to see if we could recreate the inhibition of the neutralizing antibodies. So the FDA approved the release of the neutralizing antibody as an emergency treatment for COVID patients. So we used that one, and that one actually neutralized the against the spike receptive binding domain on the COVID viruses, and we've used different dosage of it following the supplier suggestions. And what we see is on the microscopic images, we do have a dose-dependent response in infecting lung tissues, where we have a lot less infected tissues and cells in the images where you do see the treatments. When we look at the number of lentivirus foci counted per images, we do see an infection inhibition that actually match the product data sheet supplied for this by the supplier. So we do have a SARS-CoV-2 inhibition response in the actual model here. Briefly, before we finish, it's just the next step is obviously we are only working with single organ. And as I mentioned earlier, the human body is an entire connection of different organs and tissues. So we need to make it more complex to be able to have a full picture in the preclinical toolbox. So this is where multi-organ MPS come, and you would link the different models together. And by combining it with PBPK modeling, we hope to better translate in vitro data to the in vivo and to correlate that to have a really strong set of data. And this is what we are currently doing for various applications. So liver and lung are being currently used in-house to study the effect of SARS-CoV-2 infection in treatments. We are moving to liver kidney as well, and hopefully later we'll have more and more organs connected like that. And the more we know about that, the better understanding of safety and efficacy we will have before reaching the clinic. So to summarize on all of that, what I want to say is that MPS systems closely mimic the human physiology. They are better predictor of human responses to drugs than current preclinical models. They also generate better in vitro to in vivo translation. And we do believe that MPS are key to fast-track drug development and help inherently reduce the failure rates currently observed. So before I finish, I just want to acknowledge the whole team working at CN Bio, but also collaborators at MIT who did the groundwork by developing the original technology, and all of our collaborators from all the different universities and pharma companies and CROs that have worked with us throughout the years to show you those data today. So thank you for listening, and I'm looking forward to your questions.